Hi, I'm Luke Piggott, Principal Scientist at Debiopharm.
Hi, I am Esteban Rodrigo y Medio, Executive Medical Director at Debiopharm.
AACR this year, we presented the first disclosure of our clinical data of zedoresertib in combination with lunresertib in biomarker-defined populations. In this study, we pre-selected patients with CCNE1 amplification, FBXW7 mutations, or PPP2R1A mutations. These patients respond poorly to standard of care therapies and have a high clinical unmet need. WEE1 and PKMYT1 are both involved in DNA damage repair, and they allow cancer cells to survive. WEE1 acts during S phase and G2M transition, whereas the PKMYT1 protein acts just during the G2M transition. When you inhibit both these proteins, we have seen a synthetic lethal interaction between them in the context of these biomarkers that we are exploring within this study. WEE1 was identified as a synthetic lethal hit in a PKMYT1 chemogenomic screen. And when we looked at the combination of zedoresertib and lunresertib, which inhibit WEE1 and PKMYT1 respectively, we saw strong synergy between these two drugs in vitro. We saw complete regressions, regardless of the biomarker that we used, both CCNE1 AMP, also FBXW7 mutations, and we saw this across multiple different indications, including breast cancer, gastric cancer, and ovarian cancer. Collectively, this strong preclinical data led us to explore this combination in a clinical study called the MYTHIC study, which is the data that we presented this year at AACR.
So at AACR, we presented the data from the Module 4 of the MYTHIC study. In this module, we have tested zedoresertib, a WEE1 inhibitor, in combination with lunresertib, PKMYT1 inhibitor. Schedule 1, that's the one that was presented at AACR, tested zedoresertib daily doses QD up to 260 milligrams and lunresertib BID 3 days on, 4 days off at 60 or 80 milligrams. We are testing so far intermittent schedule of zedoresertib in combination with lunresertib. This data was not presented and it's currently ongoing. So one of the key messages that we got from the AACR presentation is that the combination has been well tolerated with safety profile considered as manageable and in line with its drug as monotherapy. And no patient had to discontinue treatment due to treatment-related adverse events. This combination has shown great signals of activity, especially focusing on patients with ovarian cancer, platinum-resistant or refractory ovarian cancer, and at the RP2D with CCNE1 amplification tumors, we observe up to 60% overall response, taking in consideration RECIST of C125. It's important to mention that a lot of these patients have received prior lines of therapy with platinum-based therapy, but as well, even with ADCs recently approved in these indications, such as mirvetuximab. And even that, these patients achieve responses. We are continuing going with this study. Schedule and dose optimization is ongoing. We intend to report this in a future conference once the data will be ready.
Based on these exciting clinical results, both the safety profile as well as the efficacy signals that we have observed from today, we believe that this could present a new therapeutic option for patients harboring CCNE1 amplification, FBXW7 mutation, or PPP2R1A mutations, and we are very excited to announce that this has now received Fast Track designation by the FDA.